In DNA, Kenyon encountered a molecule with a property he could not explain through natural processes. For locked securely within its double helix structure is a wealth of information in the form of precisely sequenced chemicals that scientists represent with the letters A, C, T, and G, for adenine, cytosine, thymine, and guanine. In a written language, information is communicated by a precise arrangement of letters. In the same way, the instructions necessary to assemble amino acids into proteins are conveyed by the sequences of chemicals arranged along the spine of the DNA. This chemical code has been called the language of life, and it is the most densely packed and elaborately detailed assembly of information in the known universe.
Like other scientists working on the origin of life, Kenyon realized he had two choices. Either he had to explain where these genetic assembly instructions came from, or he had to explain how proteins could have arisen directly from amino acids without DNA in the primordial oceans. And in the end, he realized he could do neither.
It's an enormous problem how you could get together in one tiny submicroscopic volume of the primitive ocean all of the hundreds of different molecular components you would need in order for a self-replicating cycle to be established. And so my doubts about whether amino acids could order themselves into meaningful biological sequences on their own without pre-existing genetic material being present just reached, I guess for me, the intellectual breaking point sometime near the end of the decade of the 70s.
As Kenyon reevaluated his theory, new biochemical discoveries further weakened his conviction that amino acids could have organized themselves into proteins.
The more I conducted my own studies, including a period of time at NASA Ames Research Center, the more it became apparent that there were multiple difficulties with the chemical evolution account. And further experimental work showed that amino acids do not have the ability to order themselves into any biologically meaningful sequences.
Faced with mounting difficulties in his own theory and a growing body of scientific data about the importance of DNA, Kenyon was forced to confront the absolute necessity of genetic information.
The more I thought about the enormous problem that all of us who worked on this field had neglected to address, the problem of the origin of genetic information itself, then I really had to reassess my whole position regarding origins.
For Dean Kenyon, a new question became the focus of his search for life's origin. What was the source of the biological information in DNA?
If one could get at the origin of the messages, the encoded messages within the living machinery, then you would really be on to something far more intellectually satisfying than this chemical evolution theory.
Yet Kenyon faced a narrowing set of options, for he realized that, as with pure chance, natural selection was also limited in its creative power. By definition, natural selection could not have functioned before the existence of the first living cell, for it can only act upon organisms capable of replicating themselves, cells equipped with DNA that pass on their genetic changes to future generations.
Without DNA, there is no self-replication. But without self-replication, there is no natural selection. So you can't use natural selection to explain the origin of DNA without assuming the existence of the very thing you're trying to explain.
Natural selection, chance, and his own theory of self-organization had all failed to explain the origin of genetic information. Now Kenyon saw only one alternative.
We have not the slightest chance of a chemical evolutionary origin for even the simplest of cells. So the concept of the intelligent design of life was immensely attractive to me and made a great deal of sense, as it very closely matched the multiple discoveries in molecular biology.
In the years since Kenyon's rejection of chemical evolution, science has revealed the details of an entire system of information processing that bears the hallmarks of intelligent design. With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.